In this lesson, we're looking at signal transduction in neurons. We've already had a look at their basic structure, and now we are trying to explain the passage of the nervous system here. All right, signal transduction, remember, is that action of processing some kind of signal or message in one form or another. And the passage of a nervous signal does exactly this, and it can transform electrical energy into chemical energy and then back again. So it is trying to do two things, and it's using the nerve cell to do this. All right, so our chemical and electrical signals alternate as signals flow between the neurons. So it doesn't matter what kind of neuron it is, it's still doing the same one. So it's taking chemicals and then turning it into electrical energy and back and forth. Doesn't matter what it is touching. Now the nerve impulse travels along the surface of the cell membrane. So this is just a cell like any other cell, and it has this biolipid layer on the outside. This is one side, this is the other. Remember, this is a 3D structure. And then it uses chemical messengers to bridge the gap between one neuron and the next. They aren't touching. There is this tiny little space in here, and they have to bridge that gap using a chemical. So as the axon membrane, right, the membrane of this long projection in the cell, it's got charged ions flowing across the membrane all the way down to change the charge on either side. So we're talking about these kinds of ions. Some are positively charged, some are negatively charged, and that is what is moving across, and that is what we consider to be our electrical signal. And it does this using just those ions, but by the time it gets down to the axon terminal, it's got to jump that gap, and these aren't going to cut it anymore. So they have to release these chemicals known as neurotransmitters in order to bridge the gap, to go over here to actually bind to receptors on the next neuron, the next domino down, and it will signal that axon change of electrical signal all the way down this one again. Now the potential of the membrane to create this electrical signal relies on the differences in chemical charge on the inside and outside of the neuron. We talk about potential as a way of describing the movement of charged particles across the membrane. It might not be that there's only positives on one side and negatives on the other. It might be that there's more positives here and less positives here, so that the difference is that one side becomes more negative. So the potential is the way we describe that, right? Electrical potential. And the ions are the atoms that have lost or gained electrons, and they're dissolved in the fluid and the extracellular fluid, and the main players we talk about are sodium and potassium here. So the membrane proteins play a really important role in this movement of ions across the membrane. And when a neuron is at rest, there is a higher concentration of positive charges outside the membrane, and the negative charges are more inside. Now this is a really complicated diagram, but it shows the 4 stages. We've got a resting potential. This is what's happening when nothing is firing, no signal is being sent just yet. And then the very first step is depolarization. Okay, and this is what is known as an action potential. This process of the changing over, so we've got sodium and potassium at play here. We've got sodium coming into the cell, then we have potassium going out of the cell, and then it is actually pumped back around so it can reset itself. So there's depolarization, then repolarization, and then pumping back to that resting potential. It is quite complicated. So in this situation, you can see nothing's gonna move yet, but then when that first depolarization acts, the voltage gated sodium channels open up, the sodium rushes inside the cell, right? This is inside the cell, and then we have the potassium is going to go outside the cell during the repolarization, okay? Eventually the body has to pump them back so it can start again. So we can see that the voltage or potential differences, right? The potential difference changes frequently during this situation. They switch over, and this is happening all the way down the cell membrane of the axon. And you can read a little bit more about what's going on here at each of these stages where they've color-coded it. So once the action potential reaches the end of the neuron, we're at those axon terminals there, it triggers the release of a chemical signal known as a neurotransmitter. And they're just sort of sitting there in nice little vesicles waiting to be used. And what they have to do is diffuse across the gap, which is the synaptic cleft here. It is a gap where only these chemicals can start to diffuse across. Those ions aren't going to work in this situation. So an action potential in the presynaptic, pre meaning before it, so it's all coming down this way, it's going to trigger these vesicles, the membrane-bound sacs containing the neurotransmitters, to merge with the cell membrane, and they're going to release, so exocytosis is helping release these neurotransmitters. They're going to diffuse across this cleft here, and they're eventually going to hit a cell, cell receptor here and bind there, which is going to keep that domino effect happening. We can see this in this nice little animation. They're diffusing across, they're hitting onto the receptors, they're binding, and that signal transduction is going to flare that action potential all the way down here, and then the neurotransmitters are pumped back into the cell, the presynaptic neuron. So we've got action potential first, and then we have chemicals changing things at the synapse because they're moving across that cleft. There's a lot of tiny little details going on here. We're using calcium ions and all these kinds of things to help, but I need you to remember there's an action potential, it's moving sodium and potassium, and there is a synapse here where the neurotransmitters are being released. There are lots of different neurotransmitters. You've probably heard of a whole heap of these. And they play different roles for different pathways that need to be signaled, and will have really specific receptors that are specific to just grabbing onto that one and then starting the pathway that's needed there. So again, this might not seem like very much, but it is quite a complex process, and it would be worth doing some reading, please, to make sure you understand what's going on, both with the action potential and with the synapse.